Main difference between prokaryotic and eukaryotic translation. Prokaryotic and eukaryotic translation are involved in the synthesis of proteins by decoding the genetic instructions carried by the mRNA. During translation, nucleated triplets known as codons on the mRNA are translated to the sequence of amino acids. Both prokaryotic and eukaryotic translation share a similar basic plan throughout the process. However, there are several differences that can be observed in this translation process. The main differences between prokaryotic and eukaryotic translation is that prokaryotic translation occurs synchronously with its transcription, whereas eukaryotic translation occurs asynchronously with its transcription. The main difference between eukaryotic and prokaryotic translation is that prokaryotic translation occurs synchronously with its transcription, whereas eukaryotic translation occurs asynchronously with its transcription. This article explains what is prokaryotic translation, its definition, process, features. What is prokaryotic translation, definition, process, and features? What is the difference between prokaryotic and eukaryotic translation? Prokaryotic translation versus eukaryotic translation. Prokaryotic transcription and translation are simultaneous processes. Eukaryotic transcription and translation are discontinuous processes. 30S and 70S, 30S and 50S, 70S ribosome, 40S and 60S, 80S ribosomes. Prokaryotic mRNAs occur in the cytoplasm. Eukaryotic mRNAs occur in the nucleus. mRNAs are unstable, live for very few seconds to 2 minutes. mRNAs are quite stable, live for about few hours to days. Performed by 70S ribosomes in the cytoplasm, performed by the 80S ribosomes attached with the ER, endoplasmic reticulum. No definite phase for the occurrence occurs in G1 and G2 phases in cell cycle. G1 and G2 phases. Cap independent initiation, cap dependent and cap independent initiation. 3 initiation factors are involved: IF1, IF2 and IF3. 9 initiation factors are involved: E1F1, 2, 3, 4A, 4B, 4C, 4D and 5 and 6. A faster process, a slower process. A single release factor is involved like ERF1. 2 released factors are involved, RF1 and RF2. Difference between prokaryotic and eukaryotic translation comparison. What is prokaryotic translation? In prokaryotes, translation is a process of simultaneously synthesizing proteins with transcription. Translation begins just after transcribing the 5' end of the gene into mRNA. Prokaryotic translation basically occurs in 3 steps: initiation, elongation, and termination. In order to initiate the translation, the 2 subunits 50S and 30S are assembled. The 3 initiation factors 50, IF1, IF2 and IF3 help to assemble the initiation complex. N-formyl methionine is the first added amino acid in translation. N-formyl methionine. GTP is used as the energy source for the peptide bond formation between the remaining and incoming nucleotides. The translation initiation factor is EF1, EFP. The selection of start codon is facilitated by the binding of the ribosome with the Shine-Dalgarno sequence. The Shine-Dalgarno sequence is a purine-rich region located upstream of the AUG start codon. This sequence is complementary to the pyrimidine-rich region located upstream of the AUG start codon. This sequence is complementary to the pyrimidine-rich region on 16S rRNA. The 16S rRNA is composed of 30S subunit. These 2 complementary nucleotides pair together, forming a double-stranded RNA structure. This pairing brings the initiation codon into a P site of the ribosome. The first amino acid binds with P site. A ribosome consists of 3 active sites: A site, P site and E site. Incoming amino acid tRNAs other than the first amino acid tRNA binds with the A site. The peptide bond formation occurs at the P site. Exit site for the uncharged tRNA is the E site. Difference between prokaryotic and eukaryotic translation. Here we could say Shine-Dalgarno sequence. Transcription initiation in prokaryotic 70S ribosome. The two elongation factors are EFG and EFTU. The translation elongation until the ribosome reaches to one of the two stop codons UAA, UGA, and UAG. Release factors other than tRNAs recognize the stop codon. The mRNA with the termination codon at A site is referred to as the termination complex. Three release factors can be identified: RF1, RF2, and RF3. RF1 and RF2 recognize the UAA, UGA and hydrolyze the ester bond in peptidyl tRNA to release the nascent polypeptide gene. RF3 catalyzes the release of RF1 and RF2. Once the new protein released, the ribosome undergoes recycling. The ribosome recycling factor and EFG are involved in releasing mRNA and tRNA from the ribosome and association of 70S ribosome into 30S and 50X subunit. IF3 releases the mRNA by replacing the deacylated tRNA when bacteria enter the stationary phase. The translation is downregulated by the dimerization of ribosome. Ribosome dimerization is facilitated by RMF, HPF, and YFIA. The ribosome dissociation factors are RSFA and XFLX. What is eukaryotic translation? Translation is the second step of eukaryotic gene expression, a separate event for eukaryotic transcription from eukaryotic transcription. Transcription and translation occur in two different compartments in eukaryotes. Therefore, the two processes cannot occur simultaneously. Eukaryotic mRNAs are monocystonic and are processed in the nucleus by adding 5' poly A tail and splicing out of introns before they are released to the cytoplasm. Ribosomal pausing also affects the translation by co-translational folding of the newly synthesized polypeptide chain in the ribosome. This process delays the translation, giving time for translation. Eukaryotic mRNA consists of 5' cap and poly A tail. Therefore, the initiation of translation occurs in two different ways: cap-dependent initiation and cap-independent initiation. During cap-dependent initiation, the initiation factors bind the 5' end of the mRNA. These initiation factors hold the mRNA in the small subunit of the ribosome. During cap-independent initiation, internal ribosome entry sites allow the ribosome trafficking to the start site by direct binding. In eukaryotes, the first binding amino acid is methionine. The 40S ribosome subunit associates with 60S subunit from 80S ribosome. Two elongation factors are involved in eukaryotic translation: EF1 and EEF2. Elongation occurs in a similar way to that of prokaryotes. Termination in the translation is also the same as in the prokaryotic system, but the universal release factor ERF1 is capable of recognizing all three stop codons. The release factor ERF3 helps ERF1 to release the polypeptide gene. Basic steps of translation are shown. Big difference: prokaryotic and eukaryotic. Main difference: prokaryotic and eukaryotic translation. So, large subunit, P site, A site, initiation, tRNA, mRNA, small subunit, peptidyl transferase, amino acid, charged tRNA, new protein, release factor, etc. Difference between prokaryotic and eukaryotic translation. Timing: prokaryotic translation, prokaryotic transcription and translation are simultaneous process. Eukaryotic translation, eukaryotic transcription and translation are a discontinuous process. Ribosomes: prokaryotic translation 30S and 50S is equal to 70S ribosome. Eukaryotic translation 40S and 60S 80S ribosome. Messenger RNA source: prokaryotic translation, prokaryotic mRNAs occur in the cytoplasm and mRNA is polycystonic. Eukaryotic translation, eukaryotic mRNAs occur in the nucleus. After post-transcriptional modifications, they are released to the cytoplasm by nuclear pores. The mRNA is monocystonic. Lifespan of mRNA: prokaryotic translation, the mRNA are unstable and live for a few seconds to 2 minutes. But eukaryotic translation, mRNA are quite stable and live for about few hours to days. Location: prokaryotic translation, this is performed by 70S ribosome in the cytoplasm. Eukaryotic translation, this is performed by the 80S ribosome attached with the ER. Then localization and the life cycle. Prokaryotic translation, there is no definite phase for the occurrence. Eukaryotic translation, this occurs in G1 and G2 phases in the life cycle, cell cycle. Sequences in the untranslated region. Prokaryotic translation, Shine-Dalgarno sequence is found in the 5' UTR, 10 nucleotide upstream to the starter codon. Then eukaryotic translation, Cossack sequence is found in the 5' UTR, few nucleotide upstream to the starter codon. Then translation initiation, prokaryotic translation, cap independent initiation. Eukaryotic translation, both cap dependent and cap independent initiation. Initiation factors, prokaryotic translation, 3 initiation factors are involved, IF1, IF2 and IF3. Eukaryotic translation, 9 initiation factors are involved, EL1, ELF1, 2, 3, 4A, 4A, 4B, 4C, 4D and 5 and A. First amino acid, prokaryotic translation, N-formyl methionine is the first amino acid added to the polypeptide chain. Eukaryotic translation, methionine. N-formyl methionine and methionine. Elongation factor, prokaryotic translation, 2 elongation factors are involved, DFG and DFTO. Then eukaryotic translation, 2 elongation factors are involved, EEF1 and EEF2. Speed, prokaryotic translation. With prokaryotic translation is a faster process which adds 20 amino acids per second. Eukaryotic translation is a slower process which adds a single amino acid per second. Fate of first amino acid. Prokaryotic translation, the formyl group is removed from first amino acid, retaining methionine in the polypeptide chain. Eukaryotic translation, the whole methionine is removed from the polypeptide chain. Release factor, prokaryotic translation, 2 released factors are involved, RF1 for UAG and UAA and RF2 for UAA and UGA. Eukaryotic translation, a single release factor is involved, ERF1. In conclusion, translation is a universal process of synthesizing proteins as the first step in gene expression. Both prokaryotic and eukaryotic ribosome encode mRNA in fundamentally similar methods. Ribosomes are the machinery in the protein synthesis. All 20 essential amino acids are shared in both prokaryotic and eukaryotic translation process. Both processes occur in the cytoplasm, completing the 4 processes: initiation, elongation, translocation, and termination. The tRNA brings the correct amino acid, allowing peptide bonds to form between 2 amino acids. The main difference between prokaryotic and eukaryotic translation is that eukaryotic translation is a simultaneous process with transcription, whereas eukaryotic translation is a separate process from its transcription.